Good morning, students. This is 11th standard biobotany video class, video 1. In this video, we are going to start the new chapter 13, unit 5, plant physiology, functional organization. The lesson topic is photosynthesis. Dear students, the lower class itself you have studied what is photosynthesis, what are all the things required for photosynthesis. What is the end product of photosynthesis? Everything you have studied in the lower classes. Now, you are going to see in a detailed manner about photosynthesis in this video. The first topic of this lesson is photosynthetic pigments. So, pigments which is responsible for photosynthesis is known as photosynthetic pigments. So, these pigments are present in the chloroplast, is a cell organelle which is present inside the cell, or photosynthetic bacteria which captures the light energy necessary for photosynthesis. So simply we can able to say that one, a pigments which is responsible for photosynthesis is known as photosynthetic pigments. So these pigments which absorb the light energy from the sunlight for photosynthetic process. The first pigment is chlorophyll. So chlorophyll which is divided into A, B, C, A3 like that. It is a green color pigment which is present in the leaf and stem of a plant. Out of these different types of chlorophyll, chlorophyll A is the primary pigment. This chlorophyll A only acts as a reaction center of photosynthesis when compared to other pigments. For example, xanthophyll, carotenoid like that. So that chlorophyll A is known as primary pigment. The important one mark question. So this chlorophyll A which absorbs the light energy from the sunlight and do the photosynthetic process. Not only chlorophyll A, the other pigments such as B, C, carotenoid, xanthophyll, phycobillions are known as helping pigments or accessory pigments. They are also absorbing light energy from the sunlight and transported to the chlorophyll A for entire photosynthetic process. The shape of the chlorophyll is tadpole shape. In Tamil it is known as Thalapirattai Vadivam. It consists of a head and a tail. Head consists of magnesium porphyrin which absorb large amount of water content, so it is known as hydrophilic head, and a tail is lipophilic tail. Lipophilic tail means it's absorb large quantity of lipid molecule. So the name of the head is porphyrin and the tail is known as pytal. So the porphyrin head consists of 4 chiral rings linked together with carbon-hydrogen bridges. Each chiral ring consists of 4 carbons and 1 nitrogen atom. So this porphyrin ring which consists of several side groups which alters the property of the pigments. Different side groups of porphyral indicates various type of chlorophyll. When coming to the pytal, it is made up of 20 carbon alcohol is attached to the carbon 7 of the pyrrole ring. And also it contains long propionic acid ester bond. A long lipophilic tail helps in anchoring chlorophyll to the lamellae. So lamellae is the tubular-like substance which are connecting the thylakoids which is present inner side of the chloroplast. So this is said to be the structure of chlorophyll. Next one, carotenoid. Other pigments except green color is known as carotenoid. Green color, see the mango. Green color contains chlorophyll pigment. Yellow color contains lutein pigment. Then reddish yellow color or orange in color, orange red, contains carotene. So these are all the different fruit color due to the different colored pigmentation. Carotenoid which are responsible for yellow to orange pigments, which consist of a chemical compound called tetrapenes. So these pigments which absorb light energy very strongly in the blue to violet region of visible spectrum. Not only that, this carotenoid which protect the chlorophyll from photooxidative damage. So photooxidative damage means due to the high temperature. That high temperature which cause damage in the leafy region, and also it decrease the formation of chlorophyll content. That is said to be photooxidative damage that is prevented by the pigment called carotenoid. Hence, this carotenoid is known as shield pigments. Dear students, 2 more question. What is shield pigments? That means carotenoid pigments which absorb strong light energy in the blue to violet of visible spectrum. Not only that, they protect the chlorophyll from photooxidative damage, so that these pigments are known as shield pigments. Then these carotenoid pigments absorb the light energy from the sunlight and transfer to the chlorophyll pigment. Almost all the carotenoid pigments contains 40 carbon atom. They are present in the ripening of fruits, floral colors, that is different color of a flower. Not only that, during autumn season, the leaf color is also changed due to the carotenoids. So carotenoids means it's collectively known as carotene and xanthophyll. So this is the shield pigment known as carotenoids. And most of the carotenoids are tetrapenes. It's already contain 40 carbon atoms, C40, and also 56 hydrogen atom. That's why they are known as tetraterpenes. So the carotene are most abundantly seen in the plant, and also it is a precursor to vitamin A. That means from this carotene only we are producing the vitamin A. Lycopene is a red color pigment which is present in the fruits of tomato, red pepper, kudamilagai, that means red color kudamilagai, and roses. So these are all the different types of carotenoids which are responsible for different color of a fruit and flowers. Next one, xanthophyll, which is responsible for yellow color pigment. Like carotene, the xanthophyll which contains oxygen, that means C40, 40 carbon atom, 56 hydrogen atom, and 2 oxygen atom. That is different from carotene and xanthophyll. Carotene is different from carotenoid. Carotenoid means it's collectively known as xanthophyll and carotene. Lutein is another pigment which is responsible for yellow color change of leaves during the autumn season. That means at the time of autumn season, the green color leaves are modified into yellow color and fall off from the trees. இலையுதிர் காலம் அப்படின்னு சொல்லுவோம். So that will be happen due to the colored pigment known as lutein, which contains the yellow color pigment. The example of xanthophylls are lutein, violozanthine, and fucobillions. xanthine. Next one, Phycobillions. So Phycobillions are the proteinaceous pigments. They are soluble in water. They do not contain magnesium and phytol tail. These phycobillions are existed in two forms. One is phycocyanin, found in cyanobacteria, responsible for blue color. Phycoerythrin, found in rhodophycean algae, that is red algae or red color pigment. So this is said to be different types of accessory pigments and chlorophyll A, which is absorbing light energy and responsible for photosynthetic process. Next, we are going to see the photosynthetic unit, or otherwise known as quantosome. So quantosome are the morphological expression of physiological photosynthetic unit located in the inner membrane of the thylakoid lamellae. So thylakoid is a stack of coins. Coin ஒன்று மேலே ஒன்று அடிக்க வச்ச மாதிரி இருக்கும். Region is known as thylakoid, which is responsible for reaction center of photosynthesis. So in this inner membrane of the thylakoid lamellae only, they contains the photosynthetic unit is called quantosome. Very, very important 2 mark question. What are quantosomes? Quantosomes are morphological expression of physiological photosynthetic unit located on the inner membrane of the thylakoid lamellae. The measurement of the quantosome is 180 Armstrong to 160 Armstrong, and the thickness is 100 Armstrong. The granular structure of quantosome was first observed by Steinman in the year 1952 under electron microscope. The granular structure means look like a soil particle or pebbles, சின்ன சின்ன கல்லு மாதிரி. Later, another 2 scientists named Park and Biggins in the year 1964, they confirmed that the granular structure which is present in the chloroplast act as a physiological unit and responsible for photosynthetic process, and they coined the term quantosome. According to them, quantosomes contains 230 chlorophyll molecules. Important 1 mark question. 1 quantosomes which are containing how many chlorophyll molecules? 230 chlorophyll molecules. A minimum number of chlorophyll and the accessory pigments act together in photochemical reaction to release 1 molecule of oxygen or to reduce 1 molecule of carbon dioxide. So that means a minimum number of chlorophyll pigment along with accessory pigments, they act together in a photochemical reaction and release 1 molecule of oxygen is the end product of photosynthesis, or to reduce 1 molecule of carbon dioxide is the reactant. Oxygen is the product, carbon dioxide is the reactant of the photosynthetic reaction. So that this quantosome constitute the photosynthetic unit. The other 2 scientists in the year 1932, Emerson and Arnold, based on their flashing light experiment, they confirmed that 2500 colored molecule of chlorophyll is required for to fix the 1 molecule of carbon dioxide. Another important 1 mark question. According to the scientist, 230 chlorophyll molecules essential for to release 1 molecule of oxygen or to reduce the 1 molecule of carbon dioxide. According to Emerson and Arnold, the flashing light experiment, they found that 2500 molecule of chlorophyll is required for the fixing of one molecule of carbon dioxide. However, the reduction or fixation of one molecule of carbon dioxide requires how many quanta of light? Means 10 quanta of light. That is very, very important 1 mark question. 1 molecule of carbon dioxide 1 by 10 quanta of light divided by 10 of how many chlorophyll pigment? 2500. So how many molecules is there? 250 molecules. Usually, the range of molecules responsible for photosynthesis is 200 to 300 is another important 1 mark question. 10 quanta light is also important 1 mark question. So that the physiological unit of photosynthesis are known as quantosomes. According to Emerson, there is no need of 10 quanta of light. 8 quanta of light is required for the release of one oxygen molecule and reduction of one molecule of carbon dioxide. So that the quantum yield is 1 by 8 or 12%. It's a very, very important 1 mark question. Please take a note in your textbook. So this is said to be photosynthetic unit or quantosome. Dear students, see the diagram I told you. A coin-shaped structure is known as thylakoid. Inner membrane of the thylakoid only, which are containing quantosomes. Thus quantosomes are which are act as the antenna molecule, which is connected to the chlorophyll B and carotenoid is a photosynthetic pigments, which are absorbing the light energy. From that, this quantosome responsible for photosynthetic process. Next, we are going to see absorption spectrum and action spectrum. So the first absorption spectrum, the term absorption spectrum refers to complete retention of light without reflection or transmission. That means a colored pigments which are absorbing light energy from the sunlight. So they are absorbing the sunlight without reflection or transmission is known as absorption spectrum. Different pigments absorb different wavelength of sunlight. When you are plotting on the graphical representation, a curve shows the amount of absorption of different wavelength of light by a pigment is called absorption spectrum. Chlorophyll A and chlorophyll absorb blue and red color sunlight. Maximum absorption of peak for different forms of chlorophyll A is 670 to 673, 680 to 683, 695 to 705. So these are all the different wavelength of a sunlight which is placed in the absorption spectrum. This chlorophyll A and B only which are absorbing blue color wavelength to red color region. So out of this different colored pigment, chlorophyll A and chlorophyll B only, which are absorbing the large wavelength of light. Particularly, chlorophyll A absorbs the wavelength 680. So it forms the P680, that is photosynthetic process of 680. Chlorophyll B, which are absorbing the sunlight, that is wavelength P700. So that, not B, chlorophyll A is also absorbing two type of wavelength, that is 680 and 700. The range is between 680 to 700 only. The chlorophyll A, which are absorbing the large amount of sunlight energy. So based upon the wavelength, the photosynthetic system is also divided into two types. One is the PS1 and PS2. That is said to be absorption spectrum. Next one, action spectrum. So different chlorophyll pigment which are absorbing the different wavelength of sunlight for photosynthesis is measured and plotting a graphical representation against a quantum yield. So the curve which are showing the rate of photosynthesis with different wavelength of light is called action spectrum. From the graph showing the action spectrum, it can be concluded that the maximum photosynthesis is taken place the region between blue and red color. The wavelength of the spectrum absorption is maximum for chlorophyll A and chlorophyll B. So the action spectrum is the instrumental discovery of the existence of two photosynthetic systems in evolving one molecule of oxygen during photosynthesis. So this spectrum is called a action spectrum. Dear students, what is absorption spectrum and action spectrum is the important 2-mark question. Not only that, they'll ask in the 3-mark question also, write a short notes on absorption spectrum and action spectrum. Dear students, see the graphical representation which are showing different colored pigment with a wavelength of a light. And also they are given the action spectrum and absorption spectrum. So the yellow color, red color, green color, blue color showing the different colored pigment. Blue color chlorophyll A, red color beta carotene, green color chlorophyll B. So these are all the different color with wavelength. Out of this, the red color, blue color only which are showing the maximum wavelength to absorb the sunlight from 680 to 700 or 600 to 700 is the rate of photosynthesis, that is absorption rate divided into, that is measured by action spectrum and absorption spectrum. Next, we are going to see immersion experiment and Hill's reaction. It is otherwise known as red drop or immersion first effect. Dear students, now only we have seen out of this different colored pigment, blue and red region only which are absorbing large amount of wavelength of sunlight. So the photosynthesis is taken place between the wavelength of 600 to 700 nanometer. After the red colored pigments, the photosynthetic rate will be decreased so that the photosynthesis ranges will be dropped after the red color pigment. That dropping is known as red drop, which was observed by scientists named immersion. See the paragraph. Immersion conducted experiment in the unicellular alga known as chlorella, which are using only one wavelength of light, monochromatic light, at a time, and he measured a quantum yield. So that he plotted the quantum yield in terms of oxygen evolution in a graphical representation at various wavelengths of light. His focus was to determine which wavelength the photochemical yield of oxygen is maximum. He found that the wavelength is 600 to 680. That's why I told you 600 to 700. Between the 600 to 680 only, the yield of the photosynthesis is very high. And also the amount of oxygen is also evolved is maximum. Then after that, suddenly a drop is taken place in the photosynthetic rate above 680 nanometer. That region is known as red drop region or red region. So the definition of red drop or emission, first effect is the fall in the photosynthetic yield beyond the red region of the spectrum is referred as red drop or emission, first effect. So next one is the emission enhancement effect. It's the important 5 mark and 3 mark question. Emission modified its first experiment by supplying the shorter wavelength of light, that is red light, along with the longer wavelength of light, that is far red light. That means the red color is divided into two based upon the wavelength. Shorter wavelength are known as PR, that is red light. Longer wavelength are known as light, that means far red light. He found that monochromatic, that is monochromatism, what is that one? They are absorbing only one molecule of one wavelength of light energy. Monochromatic light for longer wavelength, that is far red light. When supplemented with shorter wavelength of red light, enhance photosynthetic yield and recover the drop. So this enhancement of photosynthetic yield is referred to as emission enhancement effect. So he plotted as a diagrammatic representation. Left side rate of photosynthesis, then the right side, that is light exposed to the wavelength of a sunlight. So first one, up to 650 nanometer red in color. Above 650, the photosynthetic rate is false and which creates the red color region is known as red drop region. So the nanometer is for red, that means they are having the long wavelength. So when you are joining this, two of them, 650 red color short wavelength, then 710 long wavelength for red, both we are combined together. Above 710, the photosynthetic rate is dropped. It will be shown in the color is known as blue color. After that, there is no increase in the photosynthetic rate. So the range is between 600 to 700 or 680 only, showing the maximum amount of photosynthetic range and evolution of oxygen is maximum. That is said to be emission enhancement effect. See that one, the photosynthetic rate of far red light, that means long wavelength, 710, that is 10 quanta. The photosynthetic rate of red light, that is short wavelength, 650 nanometer, showing the 43.5 wavelength, quanta. So the photosynthetic rate of red and far red equal to 650 plus 710 nanometer equal to 10 plus 43.5. So that is 72.5 is the enhancement effect. Dear students, up to this we have seen different enhancement effect of emission, action spectrum, absorption spectrum, photosynthetic unit or quantosome, and photosynthetic pigments. Up to this, I conclude my video. Remaining we will see in the next video class. God bless you. All the best, children.